No, I don't look that rich.
Folks, if we get started, I am not Robert Villa.
Oh,
What?
He's at a meeting in Mexico, and he asked me to step in and kind of just make sure it got moving. And there's lots of people helping, so thank you very much. My name is Kevin Bonine, and I'm a professor here at the U of A, and I spent a lot more time engaged with THS 10, 12 years ago. But coming back because I'm teaching a version of herpetology now, and we've got some of my students here showing up. Good to see you. Yes, that's you. That's you. All right, so we have some slides that I think Robert typically shows at each of our meetings. All right, THS, you're in the right place. Latest version of the Sonoran Herpetologist is out. So take a look at that.
I asked you for that shit long ago.
Let me see if I can view that site. Hold on a second.
Why are you adding E?
I don't know.
Okay. So take a look at that. There's always great stuff in the Sonoran Herpetologist. And Maggie already mentioned this, but merchandise is available. Here are some of the prices you were looking for, Maggie. The best hat is sold out, unfortunately. I don't know if that's still true.
That's over there. You're good.
We got it.
We got it.
Okay.
they're sold out. I'll have to bring some more.
Excellent.
to
So please shop online or shop here and support the Tucson Herp Society and the work that THS does. Please also consider engaging with THS social media in the various ways that one can. I think there's a new account that is getting ramped up, right? YouTube videos, so you can find recorded talks like this one. And this one is also being recorded and it'll be put up when Robert gets to it. And our next speaker, I believe in 2 months, is that right? Every other month, Stacy Weiss?
Actually, next month. She'll be here next month.
Next month, excellent, okay, I appreciate that. That's good, Stacy Weiss, I actually was at the research station, Southwestern Research Station at the same time Stacy was working on her PhD. So I'm excited that she'll be visiting. So please come see that. And tonight we are going to hear from Derek Benson, who is a PhD student with Dale DiNardo up at ASU. But we have a short presentation before that from Rebecca Kozard. Kozad. I added an R.
Lots of people do.
Rebecca Kozad, who's going to talk to us about amphibian reptile conservation. So why don't you come on up, Rebecca, and we'll let you get started.
Don't worry, Derek. I won't pick for your one. I'll take it.
Let me make sure that we're sharing the right thing.
Cool. So I'm just here today to give you guys a little introduction to the Amphibian Reptile Conservancy and what we're doing out here in the Southwest. We are a national organization that works obviously with amphibian and reptile conservation. We've been mostly based in the eastern half of the US, mostly in the Southeast until a couple of years ago. So we're trying to build more programs out here in the Southwest. So I'm Becca, I'm the Southwest Program Coordinator. We have a couple of crews that are out here in Arizona and New Mexico. So I'm just going to talk about a little bit of that. Also, the way that ARC works, we focus more on place than anything else. So we use what we call PARCAs, priority amphibian and reptile conservation areas. So these are similar to like important bird areas. I think they're IBAs, right? So we're sort of piggybacking off of that idea to really try to get at like where can we do the most good if we have to focus resources. So the way that these end up getting made, we have like habitat suitability models that we put a lot of different factors in, including species biodiversity, also presence of like small endemic ranges or threatened and endangered species, all sorts of things, along with habitat variables. And then once we have that nice model, then we have to try to get all of the experts in herp conservation in a state together in one room at the same time and have them duke it out over where the most important places are. So this is a slightly older map. We filled in Utah and Mississippi parkas this past year, which is awesome. So we're working on it, trying to fill out all of the states. So we also have this on our website, just an interactive map in case you're like, oh, what does that cover? You can go to our website at arcprotects.org and I'll have it at the end of the show too. But we have an interactive version of this so you can go through it. The way that we use these parkas is we really try to take that landscape strategy and put it to work at the local scale. So we're doing the local implementation. These are the parkas that we have in Arizona and New Mexico. And by delineating just specific areas to work on, this usually helps us get additional buy-in for like funding sources. You know, it's a lot easier instead of being like, we want to do all of these things, be like, we're going to do these things in this specific area. Then we usually are able to get more agencies and other funding sources to be like, all right, cool, yeah, we'll do that. ARC usually, we try to also use this as a way for us to provide capacity to folks that are working in these areas already, and helping to just sort of leverage those partnerships and create new ones. And this helps us also do more long-term projects when you're able to focus into specific areas. So those are all the parkas in the States. The ones that are not green are ones that we have currently funded projects for. So we've got 5 in Arizona and 2 in New Mexico. We also have that blue one in the middle is not actually a parka, but we do have funding for it. And that's like a restoration or restoration specific. So I just threw that on there too, so you can see it. I know this is busy. This is just to give you an idea of like where those overlaps are. Obviously, this is overlapping a ton of like Forest Service land. We also, have a lot of BLM land. There's a lot of public, as well as private lands. And this having maps like this lets us go to the Forest Service and be like, hey, we want to help you and also help these, so we can focus in specific areas. Some of the things we're doing, I'll just breeze through this a little quickly. One of our main focuses is obviously inventory and monitoring programs. You can't really do a lot else without having this as a component. Some of the species we've been working with the most out here with partners like Game and Fish and the Forest Service and Fish and Wildlife and different universities like U of A is Chiricahua leopard frog. We're helping do surveys for them, as well as narrow-headed garter snake, northern Mexican garter snake, Arizona toad. We also have a big program in the boot heel doing surveys on BLM lands, which is why I've got our cute little Gila monster up there too, as well as like a host of other ones. But those are some of our top species, and I just have listed here like some of the organizations that we've already been working with. We're always looking to add more. So if you see anything on here and you're like, hey, we're interested in stuff like this too, hit me up. We love to collaborate. We also work on doing removal of invasive species. Obviously, it's a big thing out here. We focus in Arizona, they have a lot already covered by the folks that have been here, and it's an established program like U of A, Game and Fish, like they've got it. So we just help add capacity where needed. We've been doing a lot more work with that stuff in New Mexico where they don't have as much capacity. And also doing crayfish removals and monitoring, as well as invasive vegetation. So like invasive cattails, that way helps you also remove more bullfrogs and stuff. Also a big component of this is restoration. So we work with a lot of partners doing different types of wetland and riparian restoration. So this is actually a project outside of, this is a dye ranch. It's in the near Apache Stick Reeves Forest. So this is a project with Arizona Game and Fish and Bat Conservation International. So we restored a lot. We did a lot up there. I don't remember how much. I keep looking at Karen. She's one of our field biologists. I'm like, what did we do here? But this way, it helps us make the places better. So hopefully we can get, if you build it, they will come. So if you restore the wetlands, hopefully you'll be able to get more of these species back into them too. So I have a couple of slides at the end where we've got a call for volunteers for some upcoming restoration projects too. We also do a lot of outreach. THS is huge in outreach, so we love when we get to partner with them and do stuff. And at local events, I know THS is probably also going to be at the Festival of Books. We've done it, had tables at different scientific conferences where we educate folks on native herb species as well as like invasive species. This is a game where we would teach kids and adults if they were willing. They were less willing for the most part, but I always like to challenge them. So like teach them how to ID a bullfrog from a native leopard frog, and then ask them to help us scoop out little rubber ducky sized frogs that were bullfrog to help protect our pond. So we also do those sorts of things, and we're going to be partnering on doing some upcoming workshops too. And like I said, really, we're still building a lot more partnerships and trying to strengthen them out here. So especially if you're like teaching a herpetology class at U of A, we're always looking for new ways to collaborate and really try to just add capacity where we can. We're all fighting for the same thing. Like, just want to see what we can do to help. So I have my contact information here if you're interested, as well as our website and our handle for social media, which they usually have some pretty funny stuff on there. If people want to take pictures to do that. And then just real quick, you want to get a picture? Sorry. Also, you can just come ask me later. I brought some cards, but I forgot to grab more. Whoops. So we do have a couple of upcoming events happening. We're doing restoration work. The first one is in mid-March in the Palencia Mountains. So we'll be restoring 2 earthen tanks there. And we definitely need volunteers. I think they're looking to, hopefully get like 30 people a day to just head out there, because we're going to be digging out some ponds and putting in liners to help make sure that we can have water retention there. The contact for that is Paul Stone. He's a professor at the University of Central Oklahoma. So this is more specifically for snored mud turtle habitat, but also, everything's going to appreciate having more water. Second one is going to be in Arizona, up at the White Mountains Grasslands Wildlife Management Area. This is another one. It's a longer one because we're going to be restoring 6 wetlands instead, and they're going to be larger ones. So also, we're going to need a ton of folks to come out. And for this one, you can hit me up. I'm going to put together like a volunteer sign-up list. You don't have to stay out there the entire 18 days. Come out for as much or as little as you can. That's it for me.
Thank you so much.
This is stuff to share.
Yeah.
Yeah.
All right, now for our main event. And thank you all for being here. It's good to see you. Good to see some familiar faces, some folks that I haven't seen in a while. Derek Benson is a 4th-year biology PhD candidate. That means he's jumped through some hoops already. At Arizona State University in the lab of Dr. Dale DiNardo, an aspiring ecophysiologist. Benson utilizes rattlesnakes as a model system to understand how organisms respond to increasing temperatures and drier conditions. And we're feeling it this year. All right, so let's get you set up here.
One to the right, so THS. The one that's in the center.
This one right
Not
here.
that one, no, to the right, yes, this one.
Oh, sorry, you're right, I clicked on the wrong. Clicked on the wrong one. This camera right there. Okay. Thank you, Derek.
Awesome, yeah, thank you.
Yeah, I get it.
Put the lights there.
Oh, that's.
Point it the other way, right?
I'll take it. I like it.
That's it. That's it. How about like this?
Sure.
All right, well, thank you all for having me. My name is Derek Benson. Today I'm going to be talking a little bit about rattlesnake hydro-regulation and utilizing rattlesnakes more generally as model systems to understand responses to climate change. So first off, let's see if the clicker works. There we go. You might be asking yourself, who the hell is this guy? So I'm a fourth year PhD candidate at Arizona State University in the lab of Dale DiNardo. I assume some of you either know Dale or know of Dale. He is an environmental physiologist, so he tried to understand how environmental variables affect animals and how they utilize physiology to tolerate and respond to those variables. And I'm trying to follow a similar path. My driving question, kind of what I want to understand, is how species adapt to and thrive in response to a variety of environmental variables. And this could be things like extreme temperatures, whether it be warm or cold. It could be long periods without water, drought. It could be having to hold your breath for a long time, whatever it is. I'm really interested in how animals deal with what we think of as extreme challenges. Luckily for me, perhaps not so luckily for animals, climate change is rapidly changing many places around the world. So there's a big question in biology right now of how is climate change going to impact organisms around the globe. So one of the main effects of climate change is increases in temperature. Temperature is expected to increase anywhere from 2 to 9 degrees Celsius globally by the 2050s. And these changes aren't universal, right? Not all areas will be affected equally. For example, if you're more coastal, the ocean serves as a bit of a buffer, so you won't warm up as quite as much. And if you're at more extreme latitudes, you'll probably warm more than places in more medium latitudes. Like I said, so not all areas will be equally affected, and the effect this will have on species remains speculative. Some papers show that warming temperatures might actually benefit species. For example, if there's species in montane environments that are cold more often than they like to be, warming temperatures could be beneficial. But for many species in warm environments, getting warmer could prove detrimental to their long-term well-being and persistence. Now, an often underappreciated component of climate change is drought. Drought is a very common phenomenon, as I'm sure we're all aware of in Arizona. Around 2/3 of Earth's landmass undergoes around 3 or more consecutive months without rainfall annually. So many animals around the globe have had to adapt to and thrive under drought conditions. However, with climate change, models are predicting greater variability in seasonal rainfall, and in general, increasing drought frequency and duration for many areas around the globe. So right now, it all seems kind of doom and gloom, right? This does not sound good for many species, but species aren't inanimate objects. Species can respond. They have agency, just like our atrox here. You can see them huddled up under our creosote bush. So of particular interest are fast-acting mechanisms that species use. So behavior, if it gets really hot out, I'll go inside, snakes will go under their burrows. This is something that can happen within seconds. So we're really interested in how animals can use behavior to respond to environmental conditions. Physiological changes also happen very quickly. One prominent example is vasodilation or vasoconstriction. So if I go on a run, my blood vessels will dilate in my face and I'll turn all red because I'm trying to cool off. Many animals can use this strategy as well to cool off or modulate their body temperature. And then transcription of relevant gene factors acts very quickly as well. So because climate change is going to act rapidly in the grand scheme of things, we think that these fast-acting mechanisms are going to be very important for species persistence. So with this study, we're kind of setting out to try and understand how species respond to increasing temperatures and drier conditions. And firstly, to understand how they're going to respond, we need to understand how species respond. And as biologists, we often throw this term around adaptation pretty willy-nilly, but adaptation can mean many different things, and species can adapt to environments in many different ways. Like we mentioned, we have behavioral adaptations, so seeking shelter more often, and this happens very quickly. This can happen within seconds. Physiological and transcriptional adaptations happen very quickly as well. And then we get into things that take a little bit longer. So epigenetic modifications, so changes to the actual configuration of your DNA or the methylation patterns of your DNA can take minutes to hours or even weeks. So much longer for species to mount a response, but it has the benefit of being more long lasting. So it takes longer to mount a response epigenetically, but the effects can persist even throughout generations sometimes. And then finally, we have genomic changes. So changes to coding DNA. This is very slow acting. It requires generations upon generations of selection. So if species require genomic changes to respond to climate change, they may not be able to respond quickly enough to survive. They might all die out before enough genomic changes can accumulate for these species to persist. And for the sake of our study, I'll be focusing mostly on behavior and physiology, although our study does comprise all of these elements. Okay, so now we kind of understand the problem. We understand how species can respond, but how do we study this? So we can first try and set out with how species tolerate high temperatures and drought currently, and then kind of extrapolate that into the future, right? We don't have a time machine. We can't make it hotter and drier. We can't go into the future and see how species are going to respond. So we have several options. Number one is we can do a longitudinal study over many years, 30 to 40 years. But this takes a lot of time and effort. And it's really hard to get funding over that much time. And I want to cut some corners. I don't want to spend 40 years in the field to get my PhD. So we can also use a natural temperature and aridity gradient. So we can try and find some kind of range of temperatures and aridity in the field that perhaps simulate climate change and projected impacts. And finally, we can use what we call a common garden experiment. For those of you not familiar with this term, that basically means we take animals from the wild and we subject them to the same conditions so we can kind of tweak whatever variable. So for example, if we're interested in how humidity affects an animal, we can take them into the lab, same temperature, same food, same diet, and just change the humidity a little bit to see how they respond. So for our study, we opted with these two options, and I'll be talking about our natural gradients and common garden experiments later on. Okay, so speaking of later on, it's right now. So we're very fortunate in Arizona that there's a natural precipitation and aridity and temperature gradient. Going from east to west, eastern Arizona is much cooler and more moist than western Arizona. And so both of these are showing either on, what is this? This is at the right-hand side. So that is the temperature of the warmest quarter, so of the summer in Arizona. You can see eastern Arizona down by Tucson, it's a lot cooler than western Arizona. Same thing with rainfall, and the rainfall pattern is mostly driven by monsoonal rains. Historically, eastern Arizona gets pretty reliable monsoonal rains in July, August, into September, whereas western Arizona very infrequently, if ever, gets those rains. So I'll be throwing the terms west and east around a lot. What that means, what I'm referring to, are these two field sites. So western Arizona is this green field site out here. For any of you who ever have been out west, this is Quartzsite, Arizona. Booming town for about 3 months of the year until everyone leaves. And then eastern Arizona, this is out by Picacho Peak, so not too far from here, if anyone knows where Picacho Peak is. So east and west will refer to these field sites. Okay, so now we have a question, we have field sites. What animals? So forget everything about the title of my talk and all the pictures so far. We need to go through some criteria. So firstly, we need something that spans across that range of temperature and aridity. So we can't use species that are endemic to only a small portion of that gradient, so that knocks off many cool species. We need something that's trackable. So for those of you who have done radio telemetry, most of the equipment we use in the radio transmitters are about the size of a AA battery. And so for an animal to be able to have that inside of their body cavity comfortably, they need to be sufficient size. So we knock off a lot of small animals with that requirement. It needs to be fairly abundant, right? So while it'd be cool to study a lot of different colubrid snakes, they're not as abundant as many species we need. Perhaps most important, they need to be tolerant of a lengthy dehydration. So for example, humans can go about 3 days without drinking water before we suffer real problems. Laboratory mice, maybe 24 hours. And that's really not enough time to see transcriptional changes or epigenetic changes. So we need something that can survive a long time without water. We want some pairs of populations that differ and divergent. So I'll come back to this point in a little bit. It'll make more sense, trust me. And finally, we want to avoid red tape as much as possible, right? We don't want to have to jump through a bunch of hoops. Desert tortoises are awesome, but there's a lot of permits you need to work with desert tortoises. It's very hard to draw blood and convince people that you can take desert tortoises from the wild, bringing them back to the labs. That eliminates them. So we landed on rattlesnakes, particularly of the genus Crotalus. Rattlesnakes are extremely successful genus. They've colonized a variety of environments and have evolved to tolerate a wide variety of environmental variables. They're in very hot locations, very wet locations, very dry locations, very cold locations, right? So if we can somehow understand how rattlesnakes can tolerate hot and dry, as well as cold and wet, maybe we can get a better understanding of how species need to respond to climate change. All right, so almost there, almost getting through the background. Bear with me a little bit longer. I'm going to throw in some terms around that will be useful to know. So osmolality is our measure of hydration. So osmolality very simply refers to the concentration of a solute in a solution. So as a solution gets more concentrated, the osmolality goes up, and that means an animal is more dehydrated. So what we do is you draw blood, and you can measure what the osmolality of the plasma is. Dehydrated individuals have higher osmolality. I'll be talking about total evaporative water loss, and this is how much water an animal loses across its skin, so cutaneously, and also ventilatory, breathing in and out. Cutaneous evaporative water loss, again, is just water loss across the skin, and body temperature, perhaps self-explanatory, is just the internal body temperature of an organism. I'll also talk a little bit about oxidative stress. There's three main terms you need to know. So derivatives of oxygen, reactive oxygen metabolites, or DROMs. These are particles in your cells, in your body, that are very damaging. So they are highly reactive. Whatever they come into contact with, they damage. So this causes damage to tissues, to cells, to organs, and they're generally produced when we're stressed. So if I go on a marathon run, or I have a really stressful exam coming up, my body might produce some reactive oxygen metabolites, which will cause damage. Usually not a problem in the short term, but if this happens over a long period of time, we can see accelerated aging and even increased mortality. Antioxidants, I'm sure we've all heard of. Things like cranberry juice have a lot of antioxidants. They're simply sponges that kind of soak up these reactive oxygen metabolites before they have time to damage anything important. And so oxidative stress refers to the ratio of reactive oxygen species to antioxidants. And if this ratio gets thrown off, if there's too many reactive oxygen species, damage can start happening to cells, and to tissues, and to organs, and it can have severe negative health consequences for animals. I'll come back to this point. Okay, so back to Crotalus. What did we do? Well, we took 4 species of rattlesnakes, and I'll try and use scientific names, but every once in a while I might say tiger instead of tigress. But we have the Western Diamond-backed Rattlesnake, Speckled Rattlesnake, the tiger rattlesnake, and then the sidewinder. And we have a total of 6 populations. So we have east and west Crotalus atrox. So again, referring to my east and west field sites. So one near Picacho Peak and one near Quartzsite, Arizona. Now, most people think that atrox is a single species across that range, right? They're not diverged. So presumably, if we see that they respond differently to environmental variables, say they respond differently to temperature, we kind of assume that that's something they can adapt fairly quickly to, right? They're not diverged. They're pretty much the same species, so it doesn't take that long to respond to temperature increase, per se. We have an eastern Cerastes population out by Picacho Peak and a western Cerastes population out by Quartzsite. These are a little bit more diverged depending on who you talk to. Some people say they're some species, some people say they're just one single species. Whatever the case is, they are more different between east and west than atrox are. So let's say we see no difference in response to temperature in atrox, but a difference in response to temperature in Cerastes. We can think, okay, maybe these differences take a little bit more time to evolve. So it might not bode as well for climate change. And then finally, we have Crotalus tigris and Crotalus pyrrhus. Now, tigris only really exists in the more moist eastern portion of the state. They're more specialized to cooler and more moist conditions. Pyrrhus is a hot, dry specialist. It's adapted to the hotter, drier portions of the state. So presumably, we can see, if we only see differences between these 2 species, we can think these changes might take a long time to evolve. And if we don't see any changes in the other 2 population pairs, it might not bode well in terms of climate change. So this is what I mean by divergence distance. Since tigers and pyrrhus are farther apart, it gives them more time to adapt to different conditions. Okay, so we came up with three main hypotheses. So firstly, we predicted that populations predominantly found in warmer and more arid areas, so the West, will have adapted to reduce water loss and mitigate the effects of warmer temperatures on body temperature. Secondly, we thought that divergence distance would affect the degree to which individuals respond to warmer temperatures and more arid conditions. And finally, we thought that dehydration would induce oxidative stress in these animals. Okay, so what did I do? Well, we captured 13 Crotalus atrox, 7 from the Western field site and 8 from the Eastern, and 7 Crotalus pyrrhus, anywhere from March 30th to May 10th. And so these were actually caught, the Western snakes were caught in 1 year, the Eastern snakes were caught in another, but the temperature and rainfall patterns were within the norms for both year, so we're going to compare them. When we captured each individual, we took them back to the lab and implanted them with an eye button and radio transmitter. Now the radio transmitter is for tracking, so we can release the animal and find it later. The eye button is this thing that kind of looks like a watch battery, and you implant it inside of an animal, and what it does is it records ambient temperature every 15 minutes, and it stores that value. So if I held it in my hands, it'd record the temperature of my hand every 15 minutes. If I put it in a snake and release it, I can remove it 6 months later, and then I can go back and say, on August 10th at 11:59 p.m., this snake was 29 degrees Celsius. And we can do that for all of the snakes in the population to get a general sense of what their body temperature is in the wild when they're being active or not active. So we did a total of 3 captures. So drawing your attention to the graph on the right-hand side of the screen, time point 1, this is our kind of baseline. This is when we first capture animals. This is when animals have emerged a few weeks prior to this time. Presumably they're hydrated because of the spring rains that have come in. Again, this is our baseline. Time point 2, this is early to mid-June. This is during the middle portion of the hot, dry period. So animals presumably are very dehydrated. They're dealing with warm temperatures. This should give us a general sense of how animals are responding to warm temperatures and dry conditions. And then time point 3, this is where the difference between east and west kicks in. This is where monsoons come most often in eastern Arizona. So you can see that rainfall really picks up. And most years in western Arizona, monsoonal rains don't come in that often, right? So there's a prolonged drought in western Arizona, but not in eastern Arizona. So we can see how prolonged drought is affecting these animals in the west. And for each time point, we took measurements of what I've already talked about. So cutaneous and total evaporative water loss, osmolality, and then oxidative stress. So if you're astute, you might say, well, what happened to tigris and cerastes? Cerastes pose some problems. They are extremely mobile and have very high turnover rates. One of the problems with radio tracking is if you don't find an animal, well, first, you just walk around like an idiot for hours trying to find that animal. And secondly, you can't tell if it's missing because it got predated, if it decided to move miles, or if that logger died. Well, we're seeing somewhere around 60% of animal loss in the cerastes. So we kind of had to throw those animals out. We can't do a study with only a few. With tigris, we thought we were being really smart. We said, we'll go out in the monsoon season. We'll catch them when they're most abundant. We'll track them over the winter, and then when they come out in the spring, we'll get our time points. Well, they decided they wouldn't emerge until late May and early June. So our time point 1 was totally thrown off, so we couldn't do that. But I promise we catch them for the lab, so we get something out of it. And this is just a brief video. So this is a very unhappy atrox in the field. And there's me running away. Leaving it alone. Okay, so atrox, it's distributed all across Arizona. This isn't exhaustive, but in the east, at our eastern field site, it's by far the most dominant species. Depending on who you talk to, somewhere between 85 to 95% of all snakes we see, right? So it's by far and away the most dominant snake at our field site. In the west, that number drops to 5 to 10% of the snakes we're seeing, right? So it's really pushed to the edge of its range. It's not abundant at all. And this difference across this aridity and temperature gradient mimics the difference projected by climate change. So hopefully we can see how this animal is responding to hotter and drier conditions. Pyrrhus, again, is our Western specialist. It's our hot and dry aficionado. It loves those temperatures. So hopefully we can see how does an animal that's adapted specifically to these conditions deal with it. Okay, for the lab, we took 8 individuals from each population. So are all 6. We had 2 time points for evaporative water loss. We took them. I'll go through all of this in more detail in a second. So before and after dehydration, we measured evaporative water loss. We had 3 time points for oxidative stress, so before and after dehydration, and then we gave them water and waited a month, took samples again, and then we took all the measurements we've already talked about. So what this looks like plotted out is we have 8 days prior to the start of the study, we pull water from cages so they don't have access, and then 24 hours before the start of the study, we give them water. And this is because rattlesnakes are extremely lazy. If you give them water in their cage, they'll be like, I haven't drank water in 5 days, but it's all the way over there, so I'll just wait a couple more days. But if you pull water for 7 days, they will drink, right? They like to drink then. So we do this to get them all started at a relative same amount of hydration. We begin water deprivation. For atrox, they go 12 weeks without water. They do this pretty comfortably. For tigers and cerasses, they go about 8 weeks, but an amazing length of time. Certainly enough time for epigenetic changes to occur. And then we end water deprivation. We take our blood sample there. We give them water, and then a week and a month later, we draw blood again. And then before and after dehydration, we take our measurements of evaporative water loss. Okay, so one of the ways we test how dehydrated animals are, we track their weight. So if they lose about 15% of their body weight, we say, okay, that's enough. They've lost enough water. We'll move on with taking our samples. The other way is we test their righting reflex. So if you flip snakes on their back, they absolutely hate it. They'll immediately flip themselves back over. But as they get more dehydrated, they lose that righting reflex. So they start to flip over slower and slower, and eventually they won't flip over at all. So in this snake here, this is towards the end of the
This is CP13, post dehydration.
fairly slow.
Seeing if the writing
But
reflex is still inhibited.
not super fast. Again, flips itself over. but
Not
getting a lot
nearly as bad as it was
right? So
before.
we know this animal is starting to get dehydrated. We don't need to push it too hard, so we proceeded with our trial. So walking you through, this is how we measured total evaporative water loss. I promise this is not a Dutch oven. This is a very sophisticated piece of equipment. So what we do is outside of this chamber, we have a thing called a pure gas machine, and we take air and we completely dry it out. So there's 0% humidity in that air. And then what we do, we pump air through this chamber. The snake is giving off water. We can leave it there for 8 to 10 hours. It will relax. It's giving off water. Water comes back out of the chamber into this machine, and this machine tells us how much water there is in the air. And because we're pumping in completely dry air, we can tell exactly how much water that snake is losing. So then we can calculate how much water per minute a snake is losing and then correct it for snake size or snake length. This is our measurement of cutaneous evaporative water loss. So under this machine, there's about a dime-sized hole, and you press it against the skin, and it actually freezes the water in the air and weighs it. And so there's water on the surface of all of our skins. So it freezes that and measures it, and then over time, it'll freeze all the water coming across the skin. And that's a fairly constant rate. So eventually we get a flat line rate of how much water is coming across the skin for a given amount of time. So we can calculate how much water the snake is losing. All right, I've talked a lot about west and east. Here's your example. So eastern Arizona on the right-hand side, you can see Picacho Peak way in the distance. It's comparatively lush. Western Arizona, it's blasted. It looks like the surface of Mars. Unless you're out there for a specific purpose, I don't recommend walking around Quartzsite in July. It's not super fun. You won't see much. Okay, finally results. What do we see? So in the field, perhaps unsurprisingly, when it's hot and dry, animals in the field are getting dehydrated, right? So at least we know that's working. However, we don't really see any difference between the western snakes and the eastern snakes. Our western population, where it's hotter and drier, they're not getting more dehydrated. Don't really know how. I can just tell you that's what we see. In regards to cutaneous evaporative water loss, so water loss across the skin, we see this really interesting pattern where our western atrox and western pyrrhus lose very little water pretty much all times of year. Our eastern atrox, during the monsoon season, so when there's water abundant, they crank up their evaporative water loss. So they're losing a lot more water. Why they don't just conserve water all the time, I don't know. If you know, please tell me. That would be very nice to know. We see a significant population time point interaction. So again, atrox in the east are losing more water, and our western populations are more similar to each other than our eastern atrox is to our western atrox population. Over here, this is basically showing there's no relationship between how dehydrated an animal is and how much water it's losing. So if it's more dehydrated, it's not losing more or less water across its skin. There's really nothing there. What about total evaporative water loss? So this is skin plus ventilatory, so skin plus breathing. Very similar pattern. Again, in that third time point during the monsoon, our eastern atrox are losing much more water than our western atrox and our pyrrhus populations. So it seems like the western atrox have adapted just fine in the field to lose less water over in those hot, dry conditions. We also detect a difference during that second hot, dry time point through ventilatory and cutaneous water loss. Why we see this in total and not cutaneous, we're not completely sure. It could be a measurement error. It could also be differences in actual ventilatory water loss. Something for further study for sure. And I include this graph here because there is, if you look closely enough, there is somewhat of a curvilinear relationship where as animals are more dehydrated, so farther along the x-axis, they start to lose less water, which is what you would expect if you want to conserve as much water as possible when you're more dehydrated. So makes sense. All right, so I talked about body temperature. What do we see in regard to body temperature? And this is only our western populations, so western atrox and western pyrrhus. So except for late June, there's about a week or two of overlap. Our pyrrhus population is always about half a degree warmer than our atrox population. You can see this, so just to orient you, this graph on the left-hand side, this is all times of day. So I just took every time of day, every 15 minutes, averaged it out. Middle is 1 to 5 p.m. So the hottest times of day on the right-hand side is 1 to 5 a.m. So the coolest time of day. You can see pyrrhus are always just a little bit warmer than atrox. So even though on a map, on a range map, they're occupying the same area, atrox are reducing their body temperature a little compared to pyrrhus. And we think this is mostly due to microhabitat and habitat selection. Atrox, if you've ever seen Western Diamond-backs out in the field, you go out and they're mostly away from the hills in the hot, dry portions of the summer. They're seeking refuge in burrows, all of trying to stay cool. Pyrrhus are weird. So this is a video from mid-June when it's really hot and dry. And I'm tracking this snake. You can see the tracker on the ground. And I follow the snake up to the top of a hill. I'm like, all right, this thing's got eaten by an owl. It's gone. There's no way this snake's just on the top of rocks in the middle of nowhere. And sure enough, I'm looking around, middle of the night. And there's a pyrrhus just on a rock for no reason. There's no food up there. There's no bushes. There's nothing. And it's a little bit hard to tell from our video here where exactly it is. So that corresponds to this point way over here. So in terms of elevation, that's 1493 feet. You can see it's on this big hill outcrop relative to the rest of the area. And you can see those two points next to my blue arrow on the right-hand side correspond to a measurement on July 6th and a measurement on August 27th. So it moved 20 feet from that rock in a month and a half. Down here, this is the closest I got to the hills for finding Western Diamondbacks. So in early April, when those animals emerged, I saw one atrox down there before it took off and went down into the flats. Here's another example. So we have a pyrrhus smack dab in the middle of a hill. Have no idea why it's up there. Again, this is August 19th at 8 p.m. It's just hanging out on top of a hill. So we think this is what's driving the difference in body temperature. And just to prove it, here's atrox out in the flats. So in more what you think of as cooler environments, and it's about 500 feet lower in elevation where we really start seeing atrox popping up. So it seems like even though the range is technically over that, they're really not using the same habitat. All right, what really surprised us is the difference in body temperature between our atrox in the east and our atrox in the west. So after the early July, our eastern atrox are always significantly cooler than our western atrox. To give you some kind of perspective, this snake's preferred body temperature is around 29 degrees Celsius, right? So around 86, which makes sense. If you go out in the monsoon season, you'll see them out around 86 degrees. That's what they like. Out in the west, they're hitting 31, 32, 33 Celsius. We have body temperature readings of animals 37 to 38 Celsius, which is really, really high for these animals. And yeah, we're pretty amazed by it. and definitely confused, but it indicates that we can't say whether they're not choosing to go underground, they're not choosing to mitigate, or if they simply can't. All we can say is that their body temperatures are much higher than their eastern counterparts. So we got to thinking, does this really matter, right? There's a big difference in body temperature, but how relevant is it? So we thought, okay, we have our total evaporative water loss, we have these temperatures, let's test it out, let's see what happens with water loss and temperature. And unsurprisingly, and this is, so just eastern population, we took 6 snakes, tested them randomly at 6 different temperatures. And as you increase temperature, you increase water loss very gradually, and then there's this inflection point. So from 32 to 35 Celsius, water loss takes off, right? And there's this huge increase in water loss. And around 32.5 is where we're seeing those average population values in the West, but we are seeing individuals surpass 35 Celsius body temperature in the field. So this doesn't particularly bode well for the longevity of these populations in the West if these water loss values hold true for those Western populations and are relevant in the field. Okay, so what about the lab? Well, osmolality, again, we withheld water, and unsurprisingly, they got dehydrated. Just the reference point, this is about the life-threatening value for humans. Right down here is about where humans want to be. So in 3 days, we get there. Rattlesnakes go up there, they are just fine. They come right back down the next day. Sometimes you open the cage and you can't even tell they're dehydrated, and then you give them water, they drink, they're completely happy. We didn't lose any snakes because they were dehydrated. In regards to total evaporative water loss, we see a similar pattern with the field. We see our eastern snakes, our eastern atrox, losing more water than our western atrox and our western pyrrhus. So regardless of if the animals are in the same conditions in the lab, or if we just kidnap them from the field and test them really quickly, they're responding similarly to evaporative water loss, or they have similar evaporative water loss. We also detect a difference between tigris, so our eastern population, eastern specialists, and pyrrhus, our western population, on the far right hand there in green and purple. The last thing to note is when we set out, we thought, maybe these animals that are more dehydrated will lose less water. We didn't detect any effect of hydration. So before and after hydration, there was no effect on water loss. Some animals went up, some animals went down, no clear pattern. In regards to cutaneous evaporative water loss, we see a similar pattern, but not identical. Our eastern atrox are losing more water than our western atrox and pyrrhus, but there's no difference between tigris and pyrrhus. Why this is, I don't know. I'm not infallible. I'm very fallible. I could have made a mistake. It could also be due to some kind of ventilatory water loss conserving mechanism. Who knows? Something for further study. Okay, finally into oxidative stress. So I'll start throwing those terms around. I told you I'm not a masochist. I didn't just give you terms for nothing. So antioxidants in the lab, as animals get more dehydrated, we see a significant increase in antioxidant capacity. So these animals are producing antioxidants to presumably mitigate something. In the field, we see no correlation. There's just pretty much a flat line. It doesn't matter how dehydrated you are, your antioxidant concentration is basically the same. In regards to reactive oxygen species, we see the opposite. So in the field, there's an increase. As animals get more dehydrated, they have more reactive oxygen species. In the lab, there's no correlation. So seemingly completely opposite. But what matters is oxidative stress, the ratio of antioxidants to osmolality. And if you control for individual, we see a significant negative correlation. So let me just orient you real quick. This is a little bit counterintuitive. Lower on this graph, so the way you calculate stress or oxidative stress is antioxidants divided by reactive oxygen species. So a lower value means there's more reactive oxygen species, so they're less stressed. So lower value on the Y, lower value on the Y is they're more stressed. So you can see as individuals get more dehydrated, they have more oxidative stress, right? So like we thought, individuals get dehydrated, that's hard on you, it increases oxidative stress. So not good to be dehydrated if you're a rattlesnake. Okay. And just to orient you, the colors represent different time points. So after dehydration, you'd think, okay, that's or during our hot dry period, you think most animals are dehydrated, and that is the case. And they also have more stress. Okay, so I've been talking a lot. In conclusion, what should you take away from this? Well, First off, there's really big differences in body temperature between eastern and western atrox populations. However, water loss seems to be malleable and perhaps seasonally driven. We're seeing differences in the field seasonally, but we're not seeing effective hydration. So animals aren't decreasing water loss before and after testing. We see adaptations to deal with dehydration-related oxidative stress, presumably with that increase in antioxidant capacity in the lab. All we can say for certain is that animals are experiencing more oxidative stress when they're dehydrated. And this has been theorized in other animals, but only one study has shown it so far. So it's really interesting and really cool to see that even desert specialists and species that have adapted to drought still experience oxidative stress. Finally, as for climate change implications, these temperature increases don't necessarily bode well for Western populations, given how high their body temperatures in the field already are. But seasonal changes indicate that these species might be able to respond and reduce their water loss, and that at least in some kind of silver lining, many of these mechanisms they're using might be fast acting. So future directions, immediately we're going to be doing temperature-based activity estimates. So in blue here is the environmental temperature, and in orange is the snake body temperature. You can see when blue and orange trace really nicely, that animal is above ground, it's exposed to ambient temperature. And when when orange levels out and it's not changing, but environmental temperature is, that animal is presumably underground, right? So it's in a burrow. So what we can do is we can compare our populations and our species to say, all right, how often is this population in the west underground compared to this population in the east? Are they losing foraging time? Are they spending more time underground, less time? Are they up at different times of day? Hopefully we can answer all of these questions. I'm going to be looking at transcription. So how is gene expression related to seasonal changes and hydration? We're going to be doing some omics. Omics is kind of a fancy buzzword now, but we're looking at genetic changes and epigenetic changes in these species. So are there actual differences between tiger and speckled rattlesnakes in the genome that help them survive in their different environments? Also, we don't fully understand what variables are most relevant. It could be that everything is due to humidity, right? If I tested humidity instead of withholding water in the lab, we could have all the answers. It could be purely temperature-based. Who knows? So hopefully understanding what variables are most relevant in further studies. I'm also going to be testing telomere dynamics, so how actual DNA is damaged or changes in length in response to hydration. And then we're doing some immune function studies to see how these animals respond. Okay, with that, I'd like to thank the NSF, ASU Peabody Memorial Fellowship, and the ASU Terminal Research Grant for funding this research, and thank Marty Feldner, John Sloan, Mike Conroe, Sarah Hurd, and Jeff Servos for help with fieldwork, as well as Jared Crom and Dylan Padilla with help with photos. Yeah, welcome. Any questions? Yes.
Jared, could you please back up one slide?
Yes, of course.
What time duration are we talking about across the span on the X?
So each peak, the peaks in blue, those represent a single day. So this year,
Okay.
there's one day. So this is probably like 4 a.m., 5 a.m., sunrise, temperatures heat up, sun sets, temperatures go back down.
Thank you.
Yes.
Do you think the eastern atrox would have a lower survivability if it was picked up and put in the habitat of the western?
That's a good question. I would have, this is completely a guess, but I would say no. It seems like the water loss values are malleable, and let me see if I can go back.
I remember the western.
Right, so here, we don't see any difference between east and west in terms of water, right? We don't see any differences in terms of water loss. So it seems like they can keep their water loss down to similar levels. So I would predict that if they were hot and dry all the way through, they wouldn't change their water loss. And one of the things I didn't show is what is really interesting. If you look at the shed cycle of these animals, our eastern atrox, they shed in between time points 1 and 2. Our western snakes never shed atrox or pyrrhus. We don't know why, but we know that's the case. Another thing we noticed is that in east here, all of our atrox, they have their babies during the monsoon, right? They give birth, presumably because that's when the time to give birth. Resources are great. So do the Western atrox. They give birth during when the monsoon should be. So why that is, I don't know. How shed cycle affects water loss, something for future study for certain. Did that answer your question? Yeah. Okay, thanks.
I was just wondering, do you know what atrox is like biogeographically? Biogeographically, did it evolve in a monsoon dominant system?
That's a good question. So we think rattlesnakes, and if someone has more knowledge, please. I'm a little nervous because I'm a room herpetologist, but we think rattlesnakes came up from Mexico. So they came kind of up from the Americas or Central America and spread out throughout that. Yeah. So they would have had to colonize the hotter, drier conditions. Yes.
When you were looking at the body temperature between west and east atrox, did you go and actually look like, because you had the little eye tracker on them, I'm assuming, right?
Yeah.
Did you go and see if they were consistently in burrows? Was that your idea?
Yeah, so what we did is I went out about every week and found all these animals. And a lot of the time they were above ground. So I think it's, if you go out to quartzsite, like I have pictures on my phone of my car, thermostat, whatever you want to call it, and it's like 9 o'clock and it's 110 out there. And you'll test the ground temperature at 5 a.m. when it's supposed to be coolest, and it's still 90 to 92, right? So in the east, there's a lot more opportunities to cool down by going up, or maybe stay warm by staying in the west. It's just hot. It's really hot all the time. But yeah, they were up most of the times. When I went out, I caught 18 snakes during June. I went 18 for 18. All the snakes were out, which is, thank God. But yeah, we saw the same thing in the east though. So west 18 for 18, east you're like 16 for 16. They're all up during that hot dry period, which we didn't expect. Yeah.
Sure, when I see a graph like this, I usually see one scaled on the x-axis over time rather than days of the week. And I look at that and go, wow, this looks like these guys have an acritic temperature. So do you measure 24-hour temperature? Do they ever have an acritic temperature? Kind of stabilize them? Do they behaviorally thermoregulate during a 24-hour period?
Yeah, so that's a great question. So these measurements are recorded every 15 minutes. So we get this. The problem with this, and the reason I created this is the way I did, is because I had 18 snakes and I had measurements every 15 minutes for 4 months. So that's a lot of measurements. So I kind of had to average it out to plot it. Otherwise, it's a mess. But yes, as far as in the lab, we haven't done specific experiments on the preferred body. We've done a little bit. So they prefer 29. We've seen they upregulate their body temperature in response to certain variables, and they can certainly shuffle back and forth behaviorally. Yes. So I think the answer is yes. They do behaviorally thermoregulate, and we do see that in the field. Does that answer your question, or maybe I'm not understanding it?
No, it does. Boy, I tell you, that's hot. That looks like Dipsosaurus tosalis.
Yeah, it is. It is really hot. Yeah, and like I said, this isn't even capturing some of the outliers. Like the one I showed you on top of the hill, the pyrrhus, like one of its body temperatures, it was like up here. It was really, really hot. And we were shocked by it. Yes.
Do you think there could be like behavioral changes in like microhabitat selection in the atrox between the east and the west? Because I mean, it's totally, just personal experience, but like out in the more wet areas, I mean, even up in like Phoenix, I've found atrox like same exact like sites as where their pyrrhus are, like right next to each other. I found them in like weird areas that I think of as like in the mountains and hot. Like, is that areas where it's, I mean, Phoenix would be weird because it's still not that wet, but here in Tucson too, like areas where it's a little bit wetter, they're able to go to and a little bit cooler on average than quartzsite, I guess.
Yeah, so I think what you're getting at,
hot
yeah, there's more ways to survive as a Western Diamondback in the east. There's more variability in where I'm finding them. Certainly, I see them closer up to the hills. I see more variation in behavior in the east. In the west, they're pretty much all doing the exact same thing. They're down by these creosote bushes near middens, in the middens, pretty much constantly. They're not There's no variability really. They're all doing the exact same thing. And atrox aren't the most variable snake. If you go out and try and find them, they look like a little cow pie. They're sitting out, doing what they do. But in the West, they seem to be constrained and are just trying to stay alive. Yes.
Following up on that, do you get a sense of any differences in amount of movement? I mean, I know you weren't tracking them, on a daily basis, but you've got those, 15-minute temperature measurements. Do you get a sense of, is there a difference between, did some spend a lot more time hunkered down opposed to?
Yeah, so I can kind of answer that question now. So one of the undergraduates in our lab is doing the analysis of, are they actually spending more time hunkered down? What I can tell you is that the animals in the West seem to be moving much shorter distances. So in the East, when we track, after animals come up from formation, they'll warm up for a little bit and they'll take off. We have individuals that move off a mile or more. In the West, where I am, there's more rocky outcroppings nearby. So perhaps there's closer places to overwinter. But I released one animal in September, and I came back and it was 30 feet away in March, right? So they're not moving nearly as far in the West from the limited tracking I've done over the winter. Yes.
Did you use the same individuals for both the infield and the lab?
Great question. No, we didn't. So we used the same individuals for all sampling in the field, but we used completely different individuals for the lab. So yeah, completely separate. Yes.
That's a lot of data to assimilate.
Yeah, it is a lot.
So the population on the west has a higher body temperature, but the total evaporative water loss between the east and west is no different.
So the West has higher body temperature, but we're seeing also lower water loss in the West, in the lab at least. In the lab, they have lower water loss. In the field, they also have lower water loss during that third time point.
But you would think being a higher body temperature, they have a higher metabolic rate.
Exactly. What we did is we took these animals back to the lab and tested them at the exact same temperature for all animals. So even though it's warmer out there in the field during, let's go back a second. So, and I probably should clarify that more. So even though it's warmer in the West here than it is in the East, we took those animals back to the lab and it's all at 29 degrees Celsius where we tested this and we detect this difference. So even though, and that's perhaps because they're hotter, they have to preemptively lower their water loss. So then when you test them at the same temperature, our eastern snakes are losing more water. Questions? Let me see if there's any online questions or any more questions.
A quick simple one.
Yeah.
What size transmitters were you putting in your Cerastes?
Oh, Cerastes, that's a good question. Those are a lot smaller. I would say equivalent single A battery, maybe even a little bit smaller than that. Yeah, somewhere around
Yeah, they come in sizes. Usually it's like 9 grams.
It was, yeah, 9 grams. Yep.
Yeah.
Yep, and then 13 gram was atrox. So.
Yeah.
Yeah. It's the chat here.
Why not just use the smaller trackers for all of them?
Range. So larger trackers, they have greater signal. And the problem with radio tracking, when I said I'm wandering around like an idiot, if you don't pick up a signal, you have no clue where that animal is. So usually I go to where it was last seen, and the way it works is it's a lot like a metal detector. So if you point a metal detector at the ground, you kind of sweep it and you hear a beep. It's the same thing, but it's this way. So if I point this way and the snake's over here, I'll hear a very faint beep. If I point it here, it's very loud. I'm like, okay, I'm taking off that way. The snake's that way. If I don't get any signal, I have no idea where to go. So I just pick a direction, maybe start spiraling outwards. So larger ones transmit farther. So that's why we use the large. Yeah.
So when you put those eye buttons in and you put your trackers in, assuming you're making a lateral incision in the ventral side and you just put these in the peritoneal cavity. And
yeah, intraslomically. So we open them up and we stitch them to the muscle wall.
you do stitch them in.
Yep, we stitch them in because I think, and Dale, the great person to ask, because they will migrate in their body cavity if you don't stitch them in place. Yeah, so we put them pretty low, gallbladder region a little bit lower than that, past all the digestive organs. So, so let me see this question. Do you account for the effects of temperature on the absorption of preformed water on osmolality? Yeah, so one of the great question, Robert. One of the things our previous studies in Dale's lab have shown is that when snakes eat, they don't get any hydritic benefits from their meal. So when they eat a mouse, they can't get any kind of water from it. They actually get more dehydrated from digesting a meal. So they don't get any water from their meal, at least what we've seen in atrox in the lab. Yep, so they have to go, and they can go 12 weeks, 16 weeks, 20 weeks without water, but they need rain. They need standing water to hydrate. Do I know why? Man. 1, 2, left and right. That's a great question. Oh, man. No, I don't. I would have to think about that as to why they don't get a hydric benefit. I think part of it might be difference in diet, so I'm not exactly sure. So when snakes are eating rodents or things like that, they might be metabolically more intensive to break down a mammal than it is to break down an insect or a leaf, and there's not as much water stored in that regard. Yeah.
The thought on that, it might be SDA, it might be specific dynamic action, and so much energy it takes to metabolize, assimilate that meal. They have a lot of respiratory water loss system digest.
Yeah, and we actually have some preliminary data showing that these snakes are increasing their body temperature several degrees after we feed them. So they do have to bump up their body, or they choose to bump up their body temperature in response to eating. So that could be that as their metabolism goes up, they lose more water from that. Yes.
When you were choosing the individuals in the field, did you take into account like their age and size differences? Was that like a factor in what individuals you were choosing?
Yeah, so we took only adult animals. So animals basically over a certain size. You can generally tell how old animals are based to a certain extent. So if the rattle, I don't know if you're familiar with rattlesnakes, but if the rattle tapers, right? So they're born with a button, and then this basal segment of the rattle is the width of the snake, right? So as the snake grows, that basal segment will get wider and wider. So really young snakes, they kind of have like almost like a Christmas tree shaped rattle. Old snakes have much more of a just linear, they've been this shape size for a long time. So all of our snakes were adults. We didn't use sub-adults or juveniles or anything like that.
And then for the sizing, did that make a difference in their body temperature at all? Or were you shooting for rattlesnakes that were the same size consistently?
Yeah, pretty much the same size. So male and female rattlesnakes different size, especially in atrox, right? Our largest males are getting up to over a meter, which at our field site is pretty large. Females aren't getting that size, maybe really big females like 75, 80 centimeters is getting up there. But that's a great point. So I'll make that's undergood. Yes.
What about if you saw any differences between sexes? Like, did you get males and female snakes, or did you try?
Yeah, we did. We tried. So in the lab, we tried to balance it out as much as possible. With some of the species like pyrrhus and marking it up all over the place. With pyrrhus and pyrrhus, it's a little bit harder to do that, just because they're less abundant. Let's see if I can I go forward here? But yeah, we did see at least some differences in activity between males and females. We had one female that was gravid. Wait, I'm going too far. Where? What am I looking for? Okay, I'm not sure I'm going to find it right now, but yeah, we do see differences in particularly in behavior of gravid females. They're active more often. We had a bunch of females get picked off actually that were gravid just because they're out more. They're thermoregulating, trying to maintain that temperature for those developing embryos. They get picked off. So yeah.
But did you see any differences with like
With evaporative water loss? No, we didn't. So there wasn't a sex specific effect on evaporative water loss. As far as temperature, I don't have that answer.
How about we do one more? Yeah, go ahead. Last question.
How long was the lab study?
Depending on the size of the animal, so there's a pretty general correlation between size and how long they can go. So atrox were about 12 weeks, so 12 weeks without water. Sidewinders can go about 8 weeks before they start getting issued.
So, duration of the lab study.
Oh, like from when we brought them to the lab. Okay, so I would say the longest is probably 6 months. Like if we captured an animal in August or September, maybe 7. We do our test, we'd hold it, we'd feed it over the summer and over the winter, and then as soon as we could, we released it back into the wild.
Gotcha. So even though you kind of stabilized the Western atrox, did you notice any shed cycles at 29 degrees Celsius for the Western?
Yeah, they both seem to pretty much synchronize in the lab. Well, not synchronize, but they were both shedding a lot, right? Once they get water, pretty much what happened is as soon as we brought them into the lab, they got food and water. And then after the dehydration, they would all shed. So once we give them water again, they seem like, okay, now we can shed. Why that is, I don't know. Shedding is a very water intensive process, so they have to put a lot of water under their outer layer of skin.
But you weren't noticing shedding out in the field in the Western.
We didn't see any shedding. And what we did is we put a we marked the rattle, so we put paint inside of the rattle at the basal segment. And then, so if the snake sheds, we see that a new segment there. So we know that snake shed 100%. And so we're 100% certain that those snakes in the West didn't shed because that basal segment always had paint in it. It never shifted up. We're in all of the Eastern snakes. It's 100% in the East shed. 100% in the West did not shed during that, between time point 1 through 2.
But these were all adults, right?
Yes, they're all adults.
And this is over 1 year?
Yeah, one active season.
Sorry?
Yeah, 1 year, 1 year.
So youngsters probably don't have, they have a choice in it, probably have less.
Yeah, and I mean, so every once in a while we capture a female atrox, we bring her to the lab, and then she'd grab it and give birth. And both young neonates, they shed within their first couple of weeks of life, regardless of if they're in the lab or the East or West population.
All right, how about a round of applause for Derek? Great presentation, lots of fabulous information, and you fielded our questions like a PhD defense.
Hey, I get nervous when I don't get questions.
So now what happens? What does Robert do next?
Thanks, everybody. Tells them to buy some merchandise and engages in a lot of conversation.
Perfect. So engage with your neighbors, engage with our speaker, buy some merchandise, and have a great evening.
I was
Yeah. Yeah, now I know. Now I know you're.
If I had the money.
Yep, tough. My poor wallet. If there's one thing that hates me in this world, it's that.
I had to pay $15 to park. There's a chest, there's the chapter meeting here.
I was gonna invite you when I was breaking.
It's the female fraternity.
Oh.